Bom dia. Tem algumas pessoas ainda entrando, desculpa. Bem, bom dia. Meu nome é Wagner Garcia, eu sou o diretor-presidente da Plast e gostaria de agradecer a todos vocês pela presença desse evento, vocês sempre acreditando e abrilhantando o nosso evento. Um agradecimento especial gostaria de fazer ao Dr. Paulo Neufeld, que é o nosso responsável por essa abertura de parceria junto à universidade, onde a gente consegue estabelecer essa relação público-privada. Já não é o primeiro evento, nós estamos no segundo evento. Imagino que a gente está falando de um início de vários eventos que ainda estão por vir. O Dr. Paulo é uma pessoa que sempre está apoiando e incentivando esses objetivos. E gostaria também de falar dos palestrantes que nós vamos ter hoje. Teremos a abertura através da nossa palestrante internacional, que é a Delene Strunge, que vai falar sobre as cepas. Ela é representante da empresa microbiológica, a qual nós representamos aqui no Brasil. Após a palestra dela, vamos ter um coffee break às 10h30. Em seguida, continuaremos com a palestra da professora Terezinha, da USP, falando sobre monitoramento ambiental, produtos não estéreis e estéreis e controle de contaminação. Em seguida, teremos o almoço e, às 13h30, damos continuidade com a professora Marina Braga, que é pós-doutorado da USP também, vai falar sobre GPT e validação de métodos de monitoramento ambiental. Em seguida, mais um coffee break e finalizaremos com a palestra da Delene Strunge sobre Guia de Validação e Verificação e uma aula prática que deve encerrar a nossa movimentação de hoje. Essa aula prática, talvez, ela seja executada em 2 etapas. Então, no momento da execução da aula prática, para os interessados, a gente vai formar os 2 grupos e eu acredito que seja extremamente importante, porque pode fechar todo esse leque de informações que vocês vão obter hoje. Eu não vou me alongar, gostaria de agradecer mais uma vez a presença de todos, espero que vocês aproveitem e, num futuro bem próximo, espero estar presente aqui com o Dr. Paulo também, fazendo mais uma apresentação. Dr. Paulo, muito obrigado aí pela iniciativa. Conte sempre com a gente. Muito obrigado.
Bem, primeiro eu queria agradecer a presença de vocês, né? A ocorrência de vocês aqui no nosso evento. Eu tenho muita satisfação de receber as pessoas aqui. Eu acredito nessa volta do profissional para a universidade. Eu acho que isso é importante, como um recall, né? Quer dizer, tem sempre alguma coisa para ser dita, para ser apresentada, para ser inovada, e eu acho que isso é de extrema importância. A universidade, ela cumpre esse papel quando ela recebe os profissionais, quando ela faz parceria com a empresa, porque isso é um ganho, de fato, real para o conhecimento, para a melhoria das práticas, no nosso caso, laboratoriais em microbiologia. Eu acho que essas coisas eu sou muito sensível realmente a essa realidade, a essa prática de retorno do profissional e da parceria com a empresa dentro da universidade. A nossa universidade brasileira, ela não tem muito essa tradição. Mais recentemente que a gente começa a entender que a gente precisa de fato trazer o profissional, não só o estudante da graduação, não só o estudante de mestrado ou de doutorado, mas o profissional em si, né, para uma reciclagem, para uma atualização, e eu acho que isso, de um tempo para cá, tem sido considerado pela universidade. E a nossa Universidade Federal do Rio de Janeiro, que é uma grande universidade, que tem assim, uma produção científica extremamente considerável, ela também começa a entender que é importante a presença de vocês, que é importante a presença do profissional nessa relação, né, mútua de troca entre a universidade, o saber que a universidade produz, e o conhecimento e o saber também que o profissional e a iniciativa privada também produzem. Então eu queria agradecer a vocês, a presença de vocês, queria agradecer ao Wagner, a Ellen, queria agradecer a equipe toda da Plast Lab por essa parceria, né, por essa ação em conjunto, e desejar que a gente tenha assim, um encontro realmente feliz, produtivo, agradável, que a gente possa confraternizar. E falando, usando as palavras do Wagner, é uma abertura dos trabalhos para o ano de 2017. A gente espera que a gente possa estar de novo aqui, conversar e falar sobre as coisas da microbiologia, sobre as coisas do laboratório, do diagnóstico, tá bom? Então, agradeço mais uma vez.
Good morning, everyone. First, I would like to thank, is this on? Yep, okay. I would like to thank everyone for coming to this event. My name is Delene Strunge, and I am with Microbiologics. And we provide the quality control materials, hopefully all of you are using for your quality control testing. How many of you are familiar with our products and with our company? Excellent. Well, thank you for your continued support. During this presentation, first I'll start with a little history of microbiologics and a review of our products. I really encourage questions. I like to have the presentations interactive, so never hesitate to ask a question. I might not always know the answer, so if I don't, I promise we will get an answer to you. Also, what I like to do throughout the presentations is I ask trivia questions. So we can have a little fun, and with the trivia questions, I do have prizes. So this helps make sure nobody falls asleep. Okay? So we'll begin. So at Microbiologics, we were founded in 1971. We are located in St. Cloud, Minnesota. I have been with the company just over 23 years. And when I started, we were a media manufacturer, we were a testing lab, and we also provided quality control strains. So about 10 years later, we decided to solely focus on quality control and biomaterials. So that is all we do. That's our only focus. We work with, I think we're up to now about 150 distributors around the world. We sell to over 140, 150 countries around the world. Here are a couple of pictures of our facility. Up on the left is one of our quality control labs. And on the right is our shipping department and our storage area. On average, we ship out about 200 shipments a day. At Microbiologics, our mission is to provide the highest quality biomaterials for a safer and healthier world. We live by this mission every day. The employees do, our distributors do as well. As you can, we do offer, even though up here it says over 900 strains, we are well over 1,000 strains that we provide. Okay. So with that, with our commitment to a safer and healthier world, we work very hard on being the world's most trusted provider. We are very focused in customer product and design. And what that means is we know we do not provide all of the products our customers in the world need when it comes to quality control. So it's very important if there are specific strains, as long as they're biosafety levels 1 and 2, or a type of packaging that you need, it's important that you reach out to Plast Labor and let them know so they can contact us. Almost all of the products that we add to our product line is because of customer need. We have carrying experts at the ready, and that is not just at microbiologics, but that also goes to our distributors as well. Our distributors, we provide a technical training event every year at our facility, and our distributors attend that event so they have the technical knowledge and the overall microbiologics knowledge of our products. So it's very important also if you have any technical issues, any concerns, that you reach out to Plast Labor because they'll be able to help you immediately and they will have our support as well. So with controls, quality control strains, a very typical challenge we hear from our customers is that, well, you know, we don't have the money. Replenishing our quality control strains gets to be too expensive. And we understand that and we hear that. But we remind the customers that quality control is the very base of the whole foundation of the work you do in your lab. So when you take into consideration the cost of equipment, the cost of the labor, the time, making sure you're prepared for your auditors, and so on, when you add all that up, the quality control products are very minimal, but are one of the most important parts of having a successful quality control testing lab. Sorry, I need a drink of water. So use of controls in the microlab. Conditions are always changing. There are a great increase in the recalls due to contamination, and that's, you know, we're talking food, we're talking pharmaceuticals, in the clinical. Several reports of misdiagnosis. The regulating landscape is always changing, and what that means is the regulators are changing what they want to see in the laboratories. Auditors are changing. If you're in a pharmaceutical or a food lab and you export your goods outside of Brazil and, for example, to the United States or to Europe, their demands to import have to be met. So, and I will talk a little bit more about this later, but in the food, especially in the food industry, the U.S. border protection is getting very strict, and if produce or meat come into the United States, they are stopped at the border, they are inspected, and they're looking for the certification that these products were tested in a very well quality controlled environment. And if the documentation is not provided, U.S. border will refuse the shipment. I'm sorry, Mario. Personnel and lab management changes. So you have, you know, the people in your lab that have been there and know what they're doing constantly, day after day. New people come in, they might have a new way of doing quality control. The consumers and the patients all around the world are becoming very aware of the need or the reassurance that what they are putting into their body is not going to make them sick or even worse, kill them. So the stakes are high, and your brand and reputation are the biggest risk. So when you think of all that, having that proper quality control using fresh cultures is a very small price to pay for protection and a reputation of your brand. Okay. We do serve 7 distinct markets, and we are expanding. Currently, pharmaceutical, cosmetic, food, medical device, education, clinical, and water and environmental. Now, we do not grow the strains at our facility. We do obtain them primarily from ATCC, which is the American Type Culture Collection. We also obtain them from NCTC, which is in England, NCIMB, which is in Scotland, and most recently, the CDC, which is the Center for Disease Control. We provide more than 20 customer-focused formats, which we will go through those. So for our qualitative product line, we have our Quick Stick Plus, and I should say in the afternoon, we will be doing hands-on training, so you have the opportunity to work with our Quick Stick and also one of our enumerated products, the AccuShot. We have our LIFO disc, our Lab Elite, which is a certified reference material, and because we are accredited ISO Guide 34, that makes us a reference laboratory, which gives us the ability to provide certified reference material. And importantly, we also provide quality control sets and panels for identification systems. We've recently entered into many agreements with companies such as with the MALDI-TOF, of course, Vitek, Cepheid, I think BioFire, and so on. So if you're using identification systems in your lab, which pretty much everyone does, we do provide the sets that the manufacturers of these instruments recommend using to do the proper quality control. For the food industry, we provide easy food process control and an easy spore. We also have what is called our e-Power, and the e-Power can be used in a variety of ways for your testing. They're used to perform control functions for microbial detection, enumeration, equipment validation, method validation, and so on. And with our e-Power, we offer a variety of strains in a variety of concentrations between 10 to the second up to 10 to the 8th. So not all of our products come in this specific format. And for example, E. coli, we might have it available in the E2 or the E6. So sometimes if you need a higher concentration, reach out to Plast Labor, let us know. Then we also have the e-Power. So we have our regular e-Power, and then we have our e-Power certified reference material. For water testing, in the pharmaceutical, food labs, we have our Easy HydroShot. Molecular is an area that we're beginning to get into, so we are really expanding the product line for our synthetic and inactivated controls. In the pharmaceutical, we do have our Easy AccuShot, and like I said, we will do a hands-on training later with that. So we can skip that one, Mario. So with the AccuShot, quickly, it comes with 5 vials of, and five vials of a pellet, and also 5 vials of hydration fluid. With the AccuShot, once the inoculum is made, it does have 8-hour stability. So after you make the inoculum and you maybe plate out a couple of plates, then you put that back in the refrigerator, and when you're ready to take it out, you just take it out, let it warm to room temp, give it a little shake, and it's ready to go. Also on there, one more time, back. We do guarantee the AccuShot, the one-step, the Easy CFU to provide a concentration, final concentration of 10 to 100 CFU, which of course is the requirement in pharmaceutical for growth promotion testing. Our AccuShot Select is a kit, and it comes with 1 pellet of the top 5 compendial strains recommended by the pharmacopoeias. Mario, can you go back? Or back one more. No, I'm sorry, back. Rewind. One more, and one more. So on this slide, you'll see the little vials that have the blue lids. Those are the vials that contain the pellets. And you'll see one with a yellow lid. Okay, now when you're in the pharmaceutical and you're doing your testing, who wants to tell me your favorite strain to work with?
Good question.
Pseudomonas, yes. So because Pseudomonas is such a challenge, we gave it its own special color. But in reality, we have a special hydration fluid to make the inoculum for the Pseudomonas aeruginosa. It does help with the stability of that strain. So it's very important that if you're using multiple strains of the AccuShot, that the hydration fluid for the Pseudomonas is not mixed up with the other strains such as Bacillus or E. coli, etc. Okay. For the AccuShot Select, again, this is a kit. It was developed for laboratories that do very minimal growth promotion testing. And it has the same features as the Easy AccuShot. Okay. Our very first enumerated product for growth promotion testing is our Easy CFU one-step. Or no, the Easy CFU. We gotta go back. Where'd it go? Oh, it's the next one. I'm jumping ahead. I apologize. I'm so excited to be here that I'm talking ahead of myself. So I'll start with our Easy CFU. our Easy CFU was designed for laboratories that do large volumes of quality control testing. You can go to the next one on this. It does provide 90 tests per vial. Now with this, it does only offer a 30-minute stability. So once that inoculum is made, it needs to be used within 30 minutes. And there are dilution steps involved with the preparation of this product. And then if you can go back, so the next one is our Easy CFU one step. This is very similar to our Easy CFU, but it provides less amounts of testing, which is 19 inoculum per vial. This also has the 8-hour stability. I do have to go back again, sorry. Now with our Easy AccuShot, our Easy CFU, and our Easy CFU one step, how many of you are familiar with those products and use those products? Well, we have to change that, okay? So it's important for those of you that do use it, on the label of the product, we provide a mean assay value. Many customers will call us and say, microbiologics, on the label, you obtained a CFU count of 40. But in our lab, we're getting 35. So it's important that the customers recognize, and we'll get into this more during GPT. It's important that the customers know they will not obtain the same recovery as what we do in our laboratory. So something to keep in mind for the next presentation. Our Easy PEC, you can go to the next one, is a preservative efficacy control. So laboratories or manufacturers, pharmaceutical, food, that are putting preservatives into their product. This is the product they need to use to challenge the levels of those preservatives. With this product, it comes with a guaranteed recovery of 10 to the 5th to 10 to the 6th CFU, which is the requirement of USP Chapter 51. Okay, and then some new products that we've recently launched is our genomic mycoplasma. And these are used for mycoplasma testing. This is more clinical. We can kind of jump through this. And a service that we provide, and I will talk more about this afternoon, is called Custom Solutions. And what we do is we take your environmental isolate that you find in your laboratory and we preserve it for you, and we make it into a ready-to-use format that's easy and convenient. So again, we'll talk more about this later. So we can skip that because we'll talk more about that. Here's just a couple of pictures of the microbiologics customer and technical support. Again, I cannot stress enough, if you have any challenges with our products, if you come across a strain or a configuration that you need help with, please contact Plast Labor. They will be in contact with us. Okay, so does anybody have any questions about microbiologics or the products? Just the review of them? No? Yeah? Okay, so the question is, why with our easy CFU, we only offer a 30-minute stability? And the other ones we offer 8-hour, and are we going to change that? It all depends on the customers. So far, we have not had a lot of customers requesting us to change the CFU into an 8-hour stability product. The ingredients of the, how we make the easy CFU compared to the AccuShot are different. And what's different is in most of our pellets, they have a charcoal mix in there. So that can affect a little bit of the stability just for growth promotion testing. Whereas the AccuShot is a quick dissolve, and we're able to remove the charcoal and other things that we put in there to assist with the stability. But again, customer demand, if we see a lot of customers really requesting us to increase the stability of that, then that is something we would definitely look into. Okay, does that help? Okay. Okay, so now as I promised, I said I have some trivia questions to make sure everybody's paying attention. And they're very difficult questions. So when I ask the trivia question, please be sure to raise your hand. Mario will select who to answer. So the first question is, where is microbiologics located? Very good. It's a t-shirt that says, I think it's Our Cultures Rock or help spread the culture. So this little guy, he's a fan favorite. His name is Stanley Staphylococcus, and he is our microbiologics mascot. And we've had Stanley for many, many years, and then he kind of disappeared for a while, and then we brought him back. So to win a Stanley, I got to think of a good question. I have to think of a really difficult question. How many different strains does microbiologics offer? Make sure you raise your hand. And it doesn't have to be exact. What'd she say?
More than 20. More than 20.
A little higher.
Yeah. Do the question again.
Okay, so in my introduction slide, I stated that microbiologics provides over a number of strains. What was that number? There we go. Yay. Very good. Yes. I do have more Stanleys to give away, and so just to let you know. And some other prizes. So how many of you do growth promotion testing? Okay. And how many of you use either our AccuShots or CFU in your labs? Okay. And how many of you maybe use our quick sticks? No, not too many. Okay, so how many of you are in the pharmaceutical? How many work in a pharmaceutical lab? In order for you to get a Stanley, we need to get some orders from you, okay? I'm teasing. So we will talk about growth promotion testing. We will, and I know you know what growth promotion testing is and how it is to be performed, but the main focus of this is to talk about the challenges that people face in their laboratory when doing growth promotion testing. So growth promotion testing, otherwise GPT, determines the suitability of the media in the pharmaceutical tests and used by the pharmaceuticals, by the pharmacopoeias throughout the world. So the strains that we offer and the concentration of those strains meet the requirements of the pharmacopoeias, the United States, Europe, and Japanese. So for suitability media testing on non-sterile product, the type of test performed is the microbial enumeration test. And the purpose of that is to count the total number of bacteria and fungus in a product. For non-sterile products, we're testing for a specified organism. And the purpose is to look for objectionable organisms such as salmonella. And for a sterile product, the sterility test checks for the sterility of the product. So 4 rules in GPT. Number 1, test each new batch of media. Inoculate the medium with a small number of organisms. The organisms should be 5 passages or less than the reference culture. Now, with our products, for many years, our products were at least 4 passages from the original ATCC strain. However, I think within, hopefully the next month, all of our products will be 3 passages. And of course, use the organisms listed in the USP. So variations in GPT test, the organisms, the media, and the test method. Acceptance criteria for the agar. You will compare the number of colonies on the previously approved media to the number on the new media. And this is important. The number must be within the factor of 2. So for an example, the previously approved media, they achieved 40 CFU, 40 colony forming units. Now the new batch of media, to be within the factor of 2, must be between 20 and 80 CFU. If we're doing testing broth, compare the turbidity in the new medium to the turbidity in the previously approved, and that's just a matter of your sight, just looking at it. So once you've compared the turbidity, then you will plate it out onto your non-selective agar, and the results will be less than 100. So how can microbiologics help make your growth promotion testing easier in your lab? As I explained, we provide 4 different enumerated products made specifically for growth promotion testing, and they're guaranteed to deliver between 10 and 100 CFU, again, which is the requirement of the USP. It saves time. There's no need for a spectrophotometer or turbidimeter, and it eliminates all those steps of dilution, which we'll look at later. The strains are 3 passages from the reference culture. And we always provide certificates of analysis and assay with all of our products. So they can come with the shipment, or you obtain them from Plast Labor, or they're available on our website. Years ago, we used to use cardboard containers with a metal lid for the packaging of our product, and it was horrible. Our customers complained, and so we changed our packaging into this recyclable plastic container. Inside, it contains the instructions for use, and unless you have very good eyesight, I will tell you, honestly, it's a little hard to read, but we do offer our instructions for use on our website, and they are available in Portuguese as well. So again, our Easy CFU, let me go through them. The Easy CFU one-step, our Easy AccuShot, the Easy AccuShot Select Kit. And here's just a breakdown again that we've already talked about with the advantages of each of those products. So I'd say it's been about 4 years now, we hired a third-party laboratory to do a test for us to compare doing manual methods to using our Easy AccuShot. And the results of their test showed how long, when doing manual methods, how long it does take a laboratory. So they start with the spectrophotometer. It takes 18 minutes to prepare everything, 18 hours or more for the incubation process, 85 minutes of hands-on time, 20 minutes of counting the colonies. So total hands-on time for both the spectro and turbidimeter, it's a hard word, 115 minutes. Then when they used our Easy AccuShot, total time was 30 minutes. And then the total number of steps for manual methods, 21 steps compared to 11 steps with the AccuShot. And so we like to show this to have people realize how much time they can be saving in their laboratory if they're doing manual methods compared to buying a prepared, ready-to-use culture. So some of the challenges faced while doing growth promotion testing. Okay, again, the factor of 2. Per the pharmacopoeia, for solid media, growth obtained must not differ by a factor greater than 2 from the calculated value for a standardized inoculum. The value of the standardized inoculum is obtained by counting the number of colonies on the previously approved media. How many of you that are doing growth promotion testing do this? Okay, good. And then perform side-by-side testing for a direct comparison of new and previously approved media. And you're thinking, well, it's within the factor of 2, why should I do side-by-side testing? That's just another step. Well, the reason why is to eliminate the variables, so the only variable is the medium. Many variables that you want to eliminate are temperature, the atmosphere, technique, which is a big one, method, and incubation time. So here's some examples of the variables. Here's a Candida albicans. On that side, the left side, it was incubated at 25 degrees, which was the recommendation, and on the right side at 20 degrees. And after 48 hours, you'll see you have growth on the 25 degrees and no growth on the 20 degrees. So again, temperature is very important. We do provide a guideline, a technical bulletin with recommendations of temperature and incubation time for many of our products. The atmosphere, here's a Brasiliensis, taped and not taped. For broth, Bacillus, you see with a little bit of air, or with air, you'll see the growth, and with no air, no growth. Bacillus subtilis will form secondary colonies after 24 hours of incubation. And so you'll see the one circled. What happens in the labs when they're counting the colonies, for example, we've seen people that will count that as 2 colonies. It's technically only 1. The technique of how you hold the pipette can make such a difference. We had another company do a study for us on this. And it doesn't matter if you hold it at an angle. It doesn't matter if you hold it up and down. We recommend straight up and down to ensure you get all of the inoculum out of the pipette. The main technique is consistency, to always be consistent, that all the lab personnel, everybody's doing it the same way, either by side or up and down. And of course, it's important that the pipettes are always calibrated and the right, so the inoculum, the amount of inoculum is correct in the pipette. A test that we actually did at Microbiologics, we tested 7 different brands of media, manufacturers' media. We used 1 technician, 1 lady did all this with the Pseudomonas. All the variables were eliminated. The temperature was the same, atmosphere was the same, the pipette was calibrated, and so on. The angle when she pipetted these was all the same. So all the variables were eliminated, leaving only the media. So you can see with, and again, this is Pseudomonas, the difference in the brands of the media manufacturers. So they range anywhere from 57 CFUs all the way down to 10. So sometimes, I know the very first response is if a customer receives a very low count, it's the manufacturer, you did something wrong with your organism. And we understand and we respect that because, sometimes things happen. But again, the importance is removing all the variables so you only focus on the main variable, which is the testing of the media. We've had times where customers have contacted us. They've used the same media manufacturer year after year, then all of a sudden they started having problems. And we encourage them to contact their media manufacturer to see if they changed an ingredient in their media or a process. So that's something to keep in mind as well. So a challenge on achieving growth, or is there any questions on any of that so far? No? No? Okay.
Delene,
Yes.
the test with the brains, different types of the brains. How many times did you make this test? Once, repeats 3 times.
From my knowledge, we did it one time, but they're also concluding another study for on non-selective media as well.
Great.
So,
To have the statistics.
yeah, and hopefully we should have the results for that coming out very soon. Lori's finishing that up right now. So, a non-selective medium, if you encounter any situations, consider the variables. For example, were the conditions for the Bacteroides truly anaerobic? If you're having problems with broth recovery, make sure you're checking that on the agar. And if necessary, use an environmental or unusual organism, or if it's an environmental or unusual organism, they might need a special agar to grow. And we see that a lot with a lot of customers contacting us. Well, we're trying to grow this strain with a strain outside of the pharmacopoeia. And so there's special agars that we recommend that should be used. For Clostridium sporogenes, the same suspension was used to inoculate 3 types of non-selective media. And all the media were incubated anaerobically, and you can see the difference in the recovery from soybean casein to anaerobic blood to Columbia blood agar. So with growth promotion testing, to help achieve successful results, make sure when you make your inoculum, you mix it thoroughly. Use dry plates. So if you prepare your own media, make sure the plates are dry. Use the correct growth conditions. Calibrate the pipettes. Test in duplicate, sometimes triplicate. And if necessary, also test in negative control. So some best practices for pour plates: keep the melted medium at less than 50 degrees. A source has informed us that 44 to 46 degrees is best. Give the microorganisms sufficient time to grow. It can take longer for colonies to appear on pour plates compared to spread plates. And if possible, use a backlit colony counter. So with the pour plate, if the media is too hot, here's an example using the same suspension of E. coli. The medium on the left was poured at 45. The medium on the right was poured at 59. And both plates were incubated at for 48 hours. So as you can see, the 45 degree, which is the recommended temperature for pour plate, has excellent growth. And if the media, of course, is too hot, it's just going to melt everything. And the organisms will get into the liquid and there will be no growth. So takeaways: the purpose of the growth promotion test is to ensure the media used to test the products can recover the small number of organisms. Testing in parallel will help eliminate variables and use best practices to obtain accurate and consistent results. Okay, that went kind of fast. Any questions? Somebody has to have a question.
Yeah.
Just something.
She has an issue with the growth on Pseudomonas, and she's using AcuShot,
Okay.
and this is a few, and she's testing the citrimide agar, and she's trying another kind of agar, but she is not having success.
Okay.
Success in this growth. Yeah.
Yeah, no growth or
No
little
growth.
growth?
No growth.
No growth. And all the instructions are being followed and, you know, yep, everything's being followed. So what I have to recommend is that if you can contact Plast labor, we need to know which strain, well, Pseudomonas, but we need to know what the lot number is. And when our technical department receives a complaint or a concern, the first thing they do is they look to see if we've had other complaints on that same lot. Most of the time, we issue a replacement right away so it doesn't hold up the testing. And then our technical team does an investigation. So what they do is they try to copy exactly how you did your test to see what variables we can find to better assist you. So I know there's more technical information our team will need. So if you can please let Plast labor know, and then we'll look into it. And as long as our products are used as we instruct, and our products are not frozen, we 100% guarantee our products. So if there's a problem, we will replace it. Okay?
Bom dia, pessoal. Só para aproveitar a pergunta, desculpa o seu nome, Fernanda, a questão de dificuldade no crescimento, qualquer dificuldade com a cepa, é extremamente importante que vocês reportem isso para a gente, né? E tem um formulário no próprio site da MBL, como a Delene explicou, para que a gente consiga fazer toda a rastreabilidade e identificar a raiz do problema. Então, isso facilita bastante. Às vezes a cepa ela vem duplicada, vocês têm outra cepa, né? Identificou um problema logo de cara, não deixe de fazer esse reporte para que a gente tente solucionar no menor tempo possível. Ok? Era só para complementar essa informação que ela colocou para vocês.